Welcome back to Dr. Liu's VCE Biology. Today's topic is DNA manipulation techniques and applications, as presented in the 2018 VCE Biology exam. Let's learn together. If teachers, parents, or students notice mistakes in my explanations, please provide me with your feedback. Denaturation. The double-stranded DNA is heated to a high temperature, around 94 to 95 degrees Celsius, to break the hydrogen bonds and separate it into two single strands. Annealing. The temperature is lowered, around 50 to 56 degrees Celsius, allowing short DNA sequences, called primers, to bind, or anneal, to their complementary sequences on each of the single strands. Extension. The temperature is raised slightly, around 72 degrees Celsius, and a DNA polymerase enzyme synthesizes a new, complementary DNA strand, starting from each primer. Four samples of DNA were loaded into four different wells in lanes W, X, Y, and Z. A standard ladder was loaded into the well in lane S. The results of gel electrophoresis are shown below. Identify a sample to contain 100, 150, 200, and 300 BP. Refer to the results of gel electrophoresis. First, label these lengths on a standard DNA ladder, then find the respective lengths of the DNA fragments. By comparing the DNA bands to a standard ladder, the gel electrophoresis confirms that the specific pieces of 100, 150, 200, and 300 BP are found exclusively in sample Y. Because DNA fragments have a negative charge, they are repelled by the negative electrode and travel towards the positive electrode. The longer fragments move more slowly, so they remain closer to the negative electrode. Therefore, if the longest DNA is in sample X, its band would be the one closest to the negative electrode. The gel electrophoresis results show a specific band pattern in individuals 4 and 8, who have been diagnosed with Huntington's disease, HD. This pattern represents the presence of the HD allele. Since the same bands are also found in sample 6, it indicates that individual 6 likely also possesses the HD allele and is at risk for developing the disease. More than 25 years after genetically modified (GM) food first appeared, growing GM crops remains a hotly debated topic. Some people argue that GM crops are the only way to feed the growing world population and to minimize environmental harm. Other people express different views. Bt cotton is a type of cotton that contains two genes from a soil bacterium, Bacillus thuringiensis, enabling it to produce insect-resistant proteins. Australian farmers of Bt cotton use only 15% of the quantity of the insecticide that was once needed to protect their cotton crops. However, Bt cotton is not as resistant to the main insect pest of cotton crops, Helicoverpa, as it has been in the past. In Australia, Bt cotton is picked by machine, but in India, it is picked by hand. Workers in India have developed skin allergies, which have been attributed to Bt cotton proteins. Traditionally, farmers have saved money by keeping seed from one year's crop to plant the following year. However, it is illegal for farmers to keep Bt cotton seeds because these seeds have been declared the legal property of the company Monsanto. Every year, cotton farmers must buy more seeds from Monsanto. Unlike Monsanto, the company that produces the GM food crop golden rice allows farmers to replant the rice they harvested the previous year. By inserting a gene from the bacteria Erwinia euridivora and another from a daffodil, Narcissus pseudonarcissus, into white rice, scientists produced golden rice, a rice variety containing higher levels of vitamin A. People who eat golden rice avoid vitamin A deficiency. Trials conducted in several countries have shown that golden rice is safe to eat. A transgenic organism is an organism that has had genes from another species inserted into its genome. This is the case for both Bt cotton, which contains a gene from the bacterium Bacillus thuringiensis, and GM rice, golden rice, which has genes from the bacterium Erwinia eurytovora and the daffodil Narcissus pseudonarcissus. The increased crop yield of Bt cotton is a result of its built-in resistance to certain insect pests. By minimizing damage, the cotton plants can dedicate more energy to growth and bulb production. Social implications of Bt cotton: increased costs. Farmers must purchase new seeds each year, as they cannot save them for replanting. This can lead to increased expenses and less money available for other needs like food and education. Social implications of golden rice: improved health. Golden rice improves public health by addressing vitamin A deficiency, which can save lives and reduce disease, particularly in children. This, in turn, can improve community well-being and productivity. Biological implications of Bt cotton: reduced pesticide use. The plant's built-in resistance to pests reduces the need for chemical insecticides, which lowers the environmental impact of farming. Biological implications of golden rice: enhanced nutrition. Golden rice contains genes that allow it to produce beta-carotene, which the body converts into vitamin A. This directly improves the nutritional value of the rice. Social implications of Bt cotton: potential health concerns. There are concerns that the pesticide-producing protein in Bt cotton could cause skin diseases in farmers. Although this is not scientifically proven, increased income. The reduced insect damage can lead to a higher crop yield, which may increase the farmer's income and improve the standard of living. Social implications of golden rice: reduced costs. Farmers can save and replant seeds from their harvests, which can increase their profits and food security. Proven safety. The safety of golden rice has been extensively tested and proven, which is important for public acceptance and distribution. Biological implications of Bt cotton: potential ecological risks. There is a risk that the insecticide gene could be transferred to wild relatives of cotton, gene flow, or that the continuous presence of the insecticide could lead to the evolution of resistant superweeds, or reduce the number of natural predators for the pests. Biological implications of golden rice: reduced vitamin A deficiency. The increased vitamin A content helps to prevent blindness and other health issues in populations where vitamin A deficiency is common. This section of your exam focuses on key concepts in DNA techniques and genetically modified GM foods. It is worth a total of 12 marks, which accounts for 10% of the overall grade. Multiple choice questions: 4 marks. Question 28. The principles and process of polymerase chain reaction, PCR. Questions 30 and 31. The use of DNA electrophoresis to separate and identify DNA fragments based on their size. Question 34. Interpreting electrophoresis gel results, including comparing a standard DNA ladder with unknown samples to identify a potential patient with Huntington's disease, HD. Short answer question, 8 marks. Question 10. The definition of a genetically modified organism, GMO, along with a detailed analysis of the social and biological implications of both Bt cotton and golden rice. If you need my help, please add me on WeChat or WhatsApp and send me an email. Thanks for your attention. Please note Dr. Liu's VCE Physics channel, Dr. Liu's VCE Chemistry channel, and Dr. Liu's VCE Biology channel.